to speak today. The journey for AgeX has been a short one, and we haven't acquired the millions of dollars in non-dilutive funding, but we have a couple of phase ones, and I want to talk about how we got there. My journey with non-dilutive funding, as you might imagine from the intro, is much longer, going back to the late 90s, over the last couple of decades, including having my own company for about 5 years. So let me begin by first showing this slide the lawyers require to. I refer you to our website. There will be some forward-looking statements. But let me talk about a little bit of background about AgeX itself. It's part of a new wave, I believe, of companies that are now being formed over the last few years with a focus on longevity and increasing our health span. And so our mission is to apply advanced biotechnology to the problem of aging. Now, why is this a problem? Well, to me, it's obvious, and we understood this going back to the 80s when a book came out called The Age Wave. The Age Wave describes the baby boomer generation of 70 million people, and you can imagine this what they call the pig and the python moving demographically through our society. Now the baby boomer generation is reaching the oldest boomers are in their early 70s. So we're already in the crisis. We're at the beach and we can see a wall of water. It's now a silver tsunami and not an age wave. And if you look at that curve I've labeled normal aging, the problem is with normal aging, the way people age today, is that there's this slow decline. We're keeping people alive longer, but we're alive with at least one and often two degenerative diseases of aging. Diseases like cardiovascular disease, cancer, type 2 diabetes, osteoporosis, hypertension, various neurological diseases, the list goes on. And underlying all these diseases is the aging process itself. And so now there's much more resources going in, fortunately, to this problem because it needs to be solved. And so if you look at the other curve I've labeled sustainable health, that's the way we'd prefer to age, where the population can be free of decades of degenerative and debilitating disease. So how can we get there? Well, at AgeX, we believe regeneration is the key to sustainable health. And the body is at its peak of regenerative capacity in the embryonic stage. And we've done studies at the gene and transcript level that shows there's a fundamental shift in the genetic programs that are expressed in the embryo versus even the fetus. And then as you go from a fetus to an adult and an aging adult, this regenerative capacity slowly declines, and it can't keep up with the degeneration that goes on throughout life and increases as we age, and that's why we succumb to most degenerative diseases. So in lower life forms, indeed, there are some examples where this regenerative capacity is not lost. Like in flatworms, you can divide these worms into many, many pieces, hundreds of pieces, and each piece will form a new flatworm. That's because they have these programs, these embryonic programs, which know how to recapitulate the organism at any time in its lifespan. So we ask the question, can we confer this regenerative capacity from the embryo onto the aging adult? And we call that induced tissue regeneration. So there's three ways that we're attempting to do this, and the first is simply taking the cells that we can grow now in the dish. For the last couple of decades, it's been possible, due to the discovery of Jamie Thompson, of how to grow human embryonic stem cells in the laboratory. These are pluripotent cells which can divide indefinitely, so you can keep expanding them to huge quantities. And they're capable of becoming any type of cell in the body. So essentially a toolbox, a endless source of cells, young cells that can replace aging and degenerating tissues in older people. So that's the first generation of regenerative medicine initiated in the late 90s when these cells were first grown. And we're developing a couple of products which are geared towards taking these cells and transplanting them into the patient. The second generation is to not use the cells, but use the products that are made by the cells. And it's been found that the vesicles that cells secrete actually contain nucleic acids that can reprogram neighboring cells or cells at a distance. And so oftentimes the EVs produced by these cells will have activity that can stimulate regeneration. And so that would be the second generation. The third generation is to do away with the cells completely and use our knowledge that we gain from studying these embryonic stem cells and learn what the programs are and how we can modify those using drugs, either small molecules or nucleic acids, to confer this regenerative capacity and delay or cure degenerative diseases. And so at AgeX, we're focusing on two therapeutic areas, cardiovascular. We call that product VASC1. And so we have embryonic stem cell derived cells, they're endothelial cells that we can use to treat generally ischemia. And the second product is called BAT1. Those are a special kind of fat. And if you go to our website, we just published a paper and had a press release on these brown adipose tissue cells. Brown fat is a good fat. We tend to lose that as we age, and it helps normalize metabolism. And we're planning to use these cells for treating type 2 diabetes and obesity. And so, as I said before, the second and third generation would then be to use the vesicles or drugs for these indications. So now I'm going to spend a little bit of time explaining our foundational technology. It's called PureStem. PureStem was really the brainchild of our CEO, Michael West. It's going back almost 10 years to 2008, where we started this project. And it's meant to address the problem of how to scale the cells that we can make from these what we call pluripotent or all-powerful embryonic stem cells. So how do we get enough of them to treat a large patient population? How do we get them pure enough? How do we have them have a stable identity over time, over scale up in a reactor, for example? And can we get enough of them? Can we get enough different types that we could use them to apply them to various disease indications? And finally, the fact that they're young, we believe that's going to be a key advantage of these cells, that they would be highly regenerative. So just to give you a quick example of why scale is so important, this is what you could make using steel manufacturing methods in the mid-1800s. And then Andrew Carnegie came along, came along, and he developed, need some water. He developed a way to scale that get industrial capacity, and then you could make structures like this. And so then steel became the foundational material for modern civilization. We want to do the same thing for stem cells that are made from embryonic stem cells. And so get enough to treat large patient populations and do all the clinical trials necessary. And I think the scale was overlooked in the initial excitement of the discovery that we could grow these cells. The question is, how do you get them out of the laboratory? And so if you look at the cell products that are being developed now and close to being on the market, they're for very specific indications like the eye or the spinal cord, which don't require a lot of cells. And so the way people are making those cells is to use the pluripotent stem cell. That's at the base of the tree of development. And do your scaling there. The problem is those cells are very difficult to grow and difficult to grow at high capacity. The other problem with that is that once you scale at that stage, then you have to figure out a way to get your desired cell type, and no process is 100% efficient, so you're going to have to figure out how to purify away the non-desirable cells, like the early stage cells, which could cause tumors. And so this has been a very big problem in regenerative medicine and cell therapy. Our answer to that problem is to scale not at the pluripotent stem cell, at the base of this tree, but at the middle of the tree, at a stage in between pluripotency, where they can become any cell, and a fully differentiated cell. So these cells are multipotent, but they don't form tumors. They're not pluripotent. And we did this in a way where we did a very non-biased approach. It was a brute force method where we exposed the pluripotent cells to many, many different culture conditions and got what we called candidate cultures. Without knowing exactly what they were good for, we derived clones from those, which are single cells, and then exposed those single cells to many different types of media to find just the right conditions where they would scale from a single cell to, say, a roller bottle. And from that effort, we got over 200 cell lines initially, and that came out to 140 distinct cell types, which I'll describe in a minute. But I want to show you the advantage of using these very young cells. If you look at the top of that graph, when you can see that by the time you're born, you lose half of your usable telomere length. And so this illustrates the difference between somatic cells that come from an adult or even a newborn and embryonic cells. The telomeres are much longer. And therefore, the consequence of that is a very long life in culture, which we describe as population doublings. And so typically, primary cells from the body, and most stem cell companies that are not in the embryonic stem cell space or are using mesenchymal stem cells from bone marrow and other parts of the body. And the replications come out to about 40 to 50, as you can see there for adult MSCs. But our cells go typically 70, 80, and often over 100 population doublings. What that translates to, to paraphrase Carl Sagan, is you can make billions and billions of doses of cells at, say, 100 million cells per dose. And that's enough then to think about using this for treating large populations of people. So we call this it's really a way to industrialize the stem cell field. Now, I also mentioned diversity. Initially, we got 200 clones. This is the first time we did it. And then we did a microarray analysis of the expression, and you can see the diversity is very large. If you statistically analyze these cell lines, it boils down to about 140 distinct cell types. And now that's about 8 years ago. Now we have over 250 of these cell lines. And then what do you do with the cell lines? Because we didn't select them for a particular purpose. This was done in a non-biased way. So then we have to figure out what their cell fates are. And we do something called fate space mapping, where we take a panel of maybe 70 or 80 of these lines, and we push them towards a desired cell type, like brown fat or cartilage. We look for marker expression. So it's a very large project. This took place over years to get this library. But now we have multiple lines, each distinct, that can go in these different directions. And I've shown some examples of bone, cartilage, brown and white fat we can make. We can make all the vascular cell types, smooth muscle, endothelial, and parasite. And I'm going to show just some data on BAT1. As I said, the paper's been published, coming out today. You can see very uniform expression of, for example, UCP1, which is a marker specific to brown fat. And then in 2D and 3D culture, you can see we get the fat droplets, and they stain appropriately with oil red. And so also I want to mention that high stem in the 3D culture is something we own and we've licensed in from Biotime. And it's been approved for use in humans for autologous cell transfer. So our goal is to use this material then for our cell transplantation. The VASC1 is the other product. You can see functionally they make tubular networks in culture and very uniform expression of endothelial markers. And we have the same for pericytes as well as smooth muscle cells. Okay, so I want to give you a little bit of our journey. First, I'm going to talk about how we got non-dilutive funding for the second generation of VASC1, which is to use the vesicles instead of the cells. And so if you look to the right, you can see EV publications on EVs have skyrocketed in the last 10 years. The reason for that is the discovery that they can transfer nucleic acids and essentially reprogram neighboring cells. And so our premise was that adult stem cells, like MSCs, are effective in many animal models of disease. And there are also very strong publications and evidence provided that it's actually, since they don't engraft in the cells, in the animals, it's actually the paracrine effect that's making them work. And so there's really strong support for the idea, because people put the conditioned media in, and they found that the active ingredient is EVs. And so, but we saw the problem being that it's very hard to scale up adult stem cells because of their short telomeres, and also because adult stem cells are heterogeneous in nature, so they tend to drift. So this was an opportunity for us to apply pure stem technology to manufacturing EV therapeutics in the cardiovascular space. And so we did some preliminary experiments, got some data to support that pure stem cell lines could make EVs that were bioactive. The activity we screened for was angiogenesis, blood vessel formation. We have about a dozen, and I'm just showing one example. And I show we confirmed the long life in culture compared to MSCs for this endothelial cell line. And then we also compared them to MSC exosomes. So pure stem exosomes turn out to be about 6-fold more potent than adult stem cell exosomes, also lending support for the idea that embryonic cells and their cell products are more active or more regenerative. And so we proposed that we would screen and confirm this in our phase 1 study. We would select candidates, and then we test them in an animal model of ischemia. We packaged our preliminary data. We found a collaborator for peripheral artery disease, which is a complication of diabetes. And we set about to apply. And so our first phase 1 did not get funded. I think we had a moderate sort of score in the 50s. Then we brought in FreeMind, and I just want to mention that FreeMind's been a tremendous help in allowing us to really focus on the science, and they take away a lot of the administrative hassles of getting these grants done and all the little registration things that you need to keep updated. So it's definitely been worth it to bring them on. And that allowed us to keep applying almost every cycle. So for a while, and we did a revision, we addressed the comments, we still didn't get funded. I think we got a worse score the second time, which happens. It's very disappointing. Now you can apply if you don't get funding after your first revision. You can't revise again, but you can send in a new phase 1, which is interesting. So we modified our phase 1 a little bit, but really addressing the comments in the first revision. But that also did not get funded. So there was something missing. And we decided, I took a break. I went to a meeting and I met another stem cell researcher who was using MSC EVs to treat stroke. And so we changed the mechanism from SBIR to STTR, and we put him as a co-PI instead of a contract researcher, which we had in the original setup. And that got us close on the first writing. I think we got a score in the 40s. And so we addressed very carefully every single negative comment of the reviewers. And with that revision, we got a very nice score of 20. And this was also in the waiver program. So that allowed us to get $386,000 for 1 year. I'll talk about our second experience. Our company's young. It's just a year that we've been spun out. But we want to do this ITR. That's our real fundamental core technology that we're developing. And so we proposed something that's a little, it's very innovative, but it's a little out there, to do an ex vivo gene therapy on MSCs from postmenopausal women. And we understood that, according to our hypothesis, that we could change the metabolic state to make them more embryonic or more glycolytic. And this would shift the balance so that the MSC stem cells would make more bone over fat. As you age, your MSCs tend to make more fat. And so we want to rejuvenate them and have them make more bone as a treatment for osteoporosis. So that was our setup. We generated some preliminary data that we could skew MSCs using a gene-modified cell, skew them towards osteogenesis. We found a very good collaborator, an expert in osteoporosis and in MSCs, and then wrote the phase 1, and this time a very different experience. We got a 35 the first time out, which normally would require a revision, but 2018 was a good year for NIH. And also, I think, as you've heard, the program officers and directors have some discretion on what they'll fund. It isn't always the absolute number of your score. So we were very fortunate. We did get funded for a year for that. So that's our funding experience. I've been in the SBIR, the non-dilutive funding field for about 23 years, I think. I've also been on a number of study sections. Here's some of the things I've learned. Some of it's redundant, but because it's already been covered. But you have to have a product. This is a commercial endeavor. You need to have a product that will be innovative enough to be somewhat disruptive and competitive with what's out there already. And I always believe that it's best to have your product in the title. I start with the title. And the title should be the product. That's how I look at it. Second, again, this was already mentioned, but feasibility, phase 1 particularly, or your fast track, you need to show feasibility. But don't do more than you need to show feasibility, because that can also work against you. They'll say you're too ambitious, and you'll never get it done. So just do enough to show that you have your proof of concept, and they'll believe that you can go to phase 2. Collaborate to expand your expertise, and I showed how we learned that the collaborator can make a difference, and the mechanism can make a difference. So switching to SDTR for us and a co-PI, which provided more expertise and more confidence that we could actually do what we said. Scientific rigor, again, this should be a no-brainer, but and you would think this would always be the case, but as we know, much of the literature has been discovered to lack reproducibility, and so NIH is really focused on this. And as a reviewer, we look at this very seriously. So have your statistician, have your statistics explained. How did you get those significant factors in your preliminary data? It has to be as good as a publication quality now when you're applying for a grant. And also validating, especially things like antibodies. How are you going to validate it? Because antibodies can have a lot of cross-reactivity. Get to know your program officer. People have talked about that already. Be persistent. You could see that did eventually pay off for us. And finally, I want to leave you with this. I don't know if there's enough baby boomers that recognize who the gentleman on the right is, but this is Johnny Carson. He is credited with this quote. Whenever he failed at a joke, he would look at Ed McMahon and say, well, they didn't buy the premise, so they're not going to laugh at the joke. Well, it's true for many things. It's true for when you're trying to sell your company or getting a grant. The premise is essential. Doesn't mean you'll get it if you have a strong premise, but if you have a good premise, well supported by the literature, having not just one paper, but many papers in good journals, and your own data, that will not guarantee it, but if you don't have it, it doesn't matter how good your approach is or your team, you need a good premise. And finally, I'll thank my associates at AgeX and also NIH and CERM, which funded some of the early work on the PureStem technology. Thank you.
Take questions.
Yeah, all right. Is there any questions?
It's too clear.
It was too clear. Well, if you have any other questions, I'll be around and you want to talk. Yes?
Maybe just one of the EVs.
Yes.
I understood correctly, you make your EVs with cells.
Yes, the stem cells produce the EVs, yes.
Is there any possibility of synthetic EVs at some point in the future?
Yeah, synthetic EVs are called liposomes, and that's had mixed results. They're hard to make reproducible and very expensive. In fact, we got this question on one of our reviews, that we believe the natural EV, and this is what people are using now, they're using EVs either their own natural activity or loading them with specific microRNAs to help with their activity. So I think that the field is moving more towards, there's something about nature, the way it produces these vesicles that is superior to, and it's very cost effective to make them in cells. All right, thank you. I'll be around if you want to talk one-on-one.